Hi, everyone. In the last lecture, we explored how light microscopes work and why magnification and resolution are important. We learned that simply making an image larger is not enough. Scientists also need enough resolution to distinguish small structures um, and separate objects. But different microorganisms and cellular structures create very different imaging challenges. Some microbes are nearly transparent and difficult to see without staining. Others are alive and moving, meaning stains or harsh preparation techniques could damage them or alter their behavior. And some structures, like viruses, ribosomes, or cellular membranes, are simply too small to resolve clearly using visible light alone. Because of these challenges, scientists have developed many different types of microscopy techniques, each designed to solve a specific problem. Some microscopes improve contrast, some allow us to observe living cells in real time, others use fluorescent molecules to target specific organisms or cellular structures, and electron microscopes can reveal detail thousands of times smaller than what can be seen with standard light microscopy. In this lecture, we'll compare several major microscopy techniques and explore what each method is best suited for, along with the kinds of biological questions each technique can help scientists answer. By the end of this module, you should be able to compare major microscopy techniques and explain what each method is used for. Bright field microscopy is the most common type of light microscopy and is usually the first microscope technique students encounter in biology labs. In bright field microscopy, visible light passes directly through the specimen and into the objective lens to create an image. One challenge with this method is that many microorganisms are nearly transparent, making them difficult to see clearly without additional contrast. To solve this problem, samples are often stained with dyes that increase visibility by coloring cells or specific cellular structures. In this image, the bacterial cells have been stained, allowing the rod-shaped bacteria to stand out clearly against the lighter background. Without staining, many of these cells would appear faint or nearly invisible under a standard light microscope. Bright field microscopy is widely used because it is relatively simple, inexpensive, and effective for observing stained bacteria, tissues, and larger cellular structures. However, its resolution is limited by the wavelength of visible light, typically to about 200 nanometers or zero point 0.2 micrometers, meaning very small structures like viruses or ribosomes cannot usually be resolved clearly. Even with those limitations, bright field microscopy remains one of the most important and widely used tools in microbiology and biology laboratories. Dark field microscopy improves contrast by making the specimen appear bright against a dark background. Unlike bright field microscopy, where light passes directly through the sample, dark field microscopy blocks central light and only allows light scattered by the specimen to enter the objective lens. As a result, organisms that would normally be difficult to see become brightly illuminated against a nearly black background. This is especially useful for observing live, unstained microorganisms that are thin or nearly transparent. In this image, the bright spiral-shaped cells are spirochetes, a group of very thin bacteria that are often difficult to visualize with standard light microscopy. In fact, just for a cool note, these are Treponema pallidum, which are the causative agent of syphilis. Many spirochetes are only about zero point 0.1 to zero point 0.5 micrometers wide, near or below the resolution limits of traditional bright field microscopes. Dark field microscopy enhances their visibility by capturing scattered light from the cells themselves. Because staining is not required, dark field microscopy can also be used to observe living cells and microbial motility in real time. Dark field microscopy is commonly used for studying live microorganisms, thin bacterial cells, and microbial movement, particularly in cases where staining might damage or alter the specimen. Phase contrast microscopy allows scientists to observe living cells without staining them at all. This is especially important because many microorganisms are nearly transparent under a normal light microscope and can be difficult to see clearly. Phase contrast works a lot like dark field by enhancing small differences in how light passes through different parts of the cell. As light moves through structures with different densities or compositions, its phase shifts slightly. The microscope converts these tiny differences and changes in brightness and contrast, making internal structures much easier to visualize. Unlike many staining techniques, phase contrast microscopy allows cells to remain alive and relatively undisturbed during observation. This means scientists can study living microbial behavior in real time, including movement, cell division, interactions between cells. And then in this image, you can see transparent microbial cells appearing with bright edges and enhanced internal contrast against the background in the right hand panel. While in the left hand panel, it does not have the phase contrast and you do not see those structures clearly. Without phase contrast, many of these cells would appear faint or nearly invisible under standard bright field microscopy. Phase contrast microscopy is commonly used to study live microbial cells, motility, and dynamic cellular processes that cannot easily be observed in fixed or stained specimens. Fluorescence microscopy uses fluorescent dyes or proteins that absorb energy and then emit visible light. These fluorescent markers can bind to specific cells, molecules, or cellular structures. When the sample is illuminated with high energy light, the fluorescent markers glow, allowing scientists to selectively visualize particular targets. This makes fluorescence microscopy incredibly useful for identifying specific organisms or molecules within complex samples. In this image, the glowing rod-shaped cells stand out clearly against the dark background because fluorescent stains are labeling the bacteria. Although fluorescence microscopy does not usually have higher resolution than standard light microscopy, it provides something equally important: specificity. Scientists can track particular microbes, proteins, or genes in ways that would otherwise be impossible. Fluorescence microscopy is widely used in microbiology, medicine, and molecular biology research. Although light microscopy is extremely useful, it has important physical limits. Because visible light has relatively large wavelengths, some objects are simply too small to resolve clearly. Viruses, ribosomes, and very small cellular structures are often beyond the resolving power of standard light microscopes. To observe structures at even smaller scales, scientists developed electron microscopy. Transmission electron microscopy, or TEM, uses electrons instead of visible light to generate images. Because electrons have a much shorter wavelength than visible light, TEM can achieve much higher resolution than standard light microscopes. In TEM, electrons pass through extremely thin samples, often less than 100 nanometers thick. Dense structures scatter more electrons and appear darker, while less dense regions appear lighter. This allows scientists to visualize internal cellular structures that are too small to see clearly with light microscopy. For comparison, most light microscopes are limited to about 200 nanometers of resolution, but structures like ribosomes are only about 20 to 30 nanometers across. TEM can also clearly visualize many viruses and cellular membranes. TEM is commonly used to study organelles, bacteria, ultrastructures, and viruses, giving scientists a detailed view inside the cell. Scanning electron microscopy, or SEM, also uses electrons instead of visible light, but unlike TEM, SEM scans the surface of a specimen rather than passing electrons through it. As electrons interact with the surface, they produce signals that create highly detailed images with a three-dimensional appearance. This makes SEM especially useful for studying microbial surfaces, biofilms, pili, and flagella. In this image, you can clearly see the shape and surface texture of the bacterial cell, along with thin appendages extending from it. Many of these structures are far too small to visualize clearly with standard light microscopy. SEM is widely used in microbiology because it provides incredibly detailed images of external cellular structures. Different microscopy techniques solve different imaging problems. Light microscopy methods help scientists observe living cells, improve contrast, and target specific structures. But visible light has physical limits, which led to the development of electron microscopy. Together, these tools allow scientists to study life across an enormous range of biological scales, from living bacterial cells to the detailed internal structures of viruses. With that, you should now be able to compare major microscopy techniques and explain what each method is used for. Thanks for coming, and see you at the next lecture.